Okay, everybody, we're going to get started. Very unruly crowd. They're excited. Yeah, excited. Yes, exactly. I see the Allen group is kind of getting out of control here now. Okay, so welcome back. I've been reminded to mention the Wi-Fi, the UHN guest Wi-Fi if you need it. And second, if you could let us come to you with the microphone when you're asking your question, just because everything's being recorded, and if you don't talk into the microphone, then we can't actually get your question recorded. And now it's my pleasure to turn it over to Catalin Toth, who's going to moderate this next session.
Thank you, and good afternoon, everyone. So this section will have 4 speakers who will discuss various aspects of microcircuit function in human cortical neurons. And our first speaker is Dr. Karri Lamsa, who is a core group leader at the Hungarian Center of Excellence for Molecular Medicine in C.E.T.S. And he is going to talk about high excitability in human brain interneurons.
Thank you. Well, does anybody have any some tips? I thought it would be very self-explanatory, but should I actually point at this one? Parvalbumin expressing interneurons are known everywhere now. Mammalian cortex as fast electrical circuits. They are known as fast-in circuits, which they also are in human. So what does it mean in real life and in terms of numbers? Is that when we look at a presynaptic pyramidal cell that speaks to a parvalbumin expressing interneurons, you can see that the time delay from the pyramidal cell spike to delay that the interneuron actually generates an axon potential is really short. We're talking about 2-3 milliseconds. And that's something quite universal in a mammalian, fast spiking circuits. So this is just to show that what happens when the fast spiking interneuron is between 2 pyramidal cells, that you get a presynaptic pyramidal cell and postsynaptic pyramidal cell, so it operates as a disynaptic inhibitory circuit, and we're talking about the delay, which is consistently about 3 to 4 milliseconds. So they really are fast spikers also in a human. This just plot summarizes the input-output transformation delay. The delay actually from the onset of an excitatory input to a cell to the time point when the cell triggers an axon potential. And this just puts together a few neurons, which are parvalbumin cell in a mouse, and in a human you see that in a mouse we are talking about a range from 2 to maybe 3.5 milliseconds. In a human it's something very similar, not definitely much longer, maybe half a millisecond longer. Now, okay, let's thinking that the fast spiking electrical circuits are phenotypically similar in all mammalian cortex, so to speak. Now it's very interesting that actually the intrinsic electrical properties of these very cells, the parvalbumin cells, in a human and in a mouse or rat are quite different. Just to make this long story short, the major differences are that the human cells have about 1.5 to 1.8 times higher input resistance, and they also have a much lower firing threshold. So in practical terms, for those who are electrophysiologists, it means that the human fast-spiking cell has a rare base current which is 3 times smaller than the same rare base current needed in a mouse or rat interneuron, about parvalbumin interneuron. So it means that basically they need 3 times less current amplitude to trigger an axon potential in the resting conditions, and 3 times less ion transfer across the membranes. That high input resistance property, it's consistent. You can see it in the entire physiological range of membrane potential in human cells compared to mouse cells. So the input resistance is higher in human cells at negative potentials, and it's higher at potentials very close to a firing threshold. So we thought about what might be a mechanistic explanation to this, that the humans have a higher input resistance. In other words, they have a lower leaking conductance in resting conditions. And when we started looking into MRI expression profile of those cells by using patch sequencing or Allen Institute database, and in addition when we did various type of immunohistochemical analysis of the actual potassium channel localization in these neurons, we couldn't really find any magic bullet explanation qualitatively why the human neurons would be higher impotence resistance neurons than a mouse neurons because of the qualitative differences of the potassium channels in the membrane. So at this stage, we just conclude that the most likely explanation to this difference is simply that the human neurons have a lower density of ion channels in their membrane. The second difference is a lower firing threshold in human cells, and it means that about 7 millivolts closer to resting membrane potential. So this is just the data we put together from about 100 PV cells in a human and the same number in a mouse, and you can see that although the range is very large in both populations, the median difference is very, very clear. So what explains mechanistically this lower firing threshold in humans? There's one very clear answer. We finally figured it out. And it is that the human neuron axon initial segment, it actually lacks Kv 1.1 and 1.2 channels. What is this channel? It is potassium channels which is activated by moderate depolarization, and its activation actually kind of hyperpolarizes the cell or resists the membrane potential depolarization. So if they are present there, as they are by literature in the mouse and rat neuron initial segments, the presence really increases the firing threshold. It makes it further away from the membrane potential. So what we did is that we used immunohistochemistry for Kv 1.1 together with parvalbumin and beta-phospectrin, which actually visualizes the axon initial segment, and did a high-resolution 3-dimensional scans and used the reference normalized immunointensity analysis, which I can tell you more about later on. But just very shortly, that actually showed that in the initial segment, which is shown here from a proximal part to a distal part, the Kv 1.1 channel is virtually lacking in the human, whereas in a mouse, it is very robust. So another Kv1 type channel that exists in PV cells in a mouse and in a rat is Kv 1.2, and very similar to Kv1, it's actually it's not present at all in a segment of human, but it seems to be moderately present in a mouse in a segment. And this protein localization and intensity data quite well actually matches with the messenger RNA expression profile. Like here we can see our own patch sequence data sets, where over here we have cells, individual cells, showing that these PV cells, or they actually lack the gene encoding the KB1.1, and some of them do have some KB1.2, whereas in mouse it's more regular. And now when we look at the alanine database, the set, which actually gives over here a transcriptomically identified PV cell type, and then over here the intensity coding shows an average or median value of the TPM values in about tens or hundreds of cells. We can see that still the KB1.1 expression gene is lacking, and it's very moderate for KB1.2 in a human, whereas in mouse it's really, really strong. So all these data are actually quite nicely in line with our molecular findings. So luckily these days you can buy drugs which have a quite a nice selectivity for Kv on potassium channel subtypes, like this dendrotoxin K, which as 100 nanomolar is selective for the Kv1.1 subtype of potassium channels. This is now the channel which really makes a difference between those two cells. And you can see that if you're watching in the drug, then there's basically no effect on the oxygen potential firing threshold in human at all. But in a mouse, it causes a massive drift. And now it's very interesting that in a mouse, it takes the value very close to the human cell value. It's about 7 millivolts. And accordingly, not only the action potential threshold is changed, also the rare-based current is changed. So in a mouse, in this situation, you need much less current to elicit an action potential when the Kv channels are blocked. This slide just summarizes what I said. The action potential threshold difference comes from the fact that human cells, they actually lack the Kv1 and 1.2 channels, which are very prominent in a mouse in this segment. Of course, we didn't bump onto this feature just as a first thing. It was probably the last thing we tested because we tried so many other analyses to explain this difference first. And I just show them very quickly here, like the expression and the intensity of the NaV1.6 low threshold sodium channel is equally robust, and they are evenly distributed in both species along the initial segment in human and in a mouse. We also looked into some geometric properties like the axon initial segment, cross-axonal diameter, and we obviously found a lot of cell-to-cell variability with this parameter, but it failed to explain the species difference. Also, we found that in the human cells, when you do the three-dimensional scannings of the initial segment length, we found that the initial segment is about 20% longer than in a mouse, and it failed to explain the species difference in the action potential firing threshold. So this all, I've been talking about mechanisms and so on, but actually it takes us to a paradox. And the paradox is that although for some obviously very relevant reason, the human cells have evolved with higher input resistance. We don't know what this reason is, but it has to be something really meaningful and important so that it has an evolutionary pressure for cells to develop it. But it has to pay a high price. So when the input resistance becomes longer, it kind of linearly, in a linear relation, it slows down the electrical temporal reactivity of the cell soma. In other words, when the cell input resistance becomes higher, the cell's temporal capacity to generate membrane potential changes, becomes much slower. So this is just what I summarized. And I go further on. This slide is probably the most important slide of the talk. So anybody who's been sleeping after the lunch, now wakey. So we found a very tight association between these two properties. And this is now very interesting because we found in mouse data and in human data, and especially when we pool these both cells, that those cells which are slowest in their soma to generate membrane potential deflections, they have the lowest firing threshold. Okay, So now it's getting very interesting. What could this mean? Because it must have developed for something. And I'm actually putting it together in this one slide. This association basically allows, we claim, it allows the slow cells to make the input-output transformation a little bit shorter. Let's get back to the first slide where I say that it's a universal profile of the parvalbumin interneuron established inhibitory circuits, that they operate very precisely with a high fidelity and with a short input-output transformation. And then we got this phenomenological difference that the human cells actually have a much slower electrical soma. So we did an experiment with dynamic clamp where we, by pharmacological tools like elongated the membrane time constant, so the electrical reactivity time course in the soma about 30%. And we found out that with all the firing probabilities of this neuron, the action potential delays, the input-output transformation delay, became elongated. Now, if we're looking into mouse cell and the human cell, so the starting point is that the mouse cells have a high input resistance, but their soma is faster. Human cells have a lower firing threshold, but the soma is slower. And we look at the dynamic clamp experiments here that if we apply here an EPSC amplitude of this much, we start with the firing threshold and then we increase the amplitude. So what is the input-output delay of this circuit? Then when we apply these same currents in a human, we find that actually, even if they have a lower soma, they manage to develop the input-output transformation with a similar or even faster delay than the mouse cells, just because, as we saw here in the model, that when we lower the firing threshold, the cell triggers an action potential at the earliest stage of the rise phase of the EPSP. Oh, I forgot I put there some errors. So this is the summary of the talk. So human cell interneurons, they develop high input resistance. We don't know why. We can speculate that most intuitively it would probably be an energy issue. We remember that if this cell type is one of the most active neuron types in the cortex. It's highly energy consuming. In the human supracranial layer, the proportion to pyramidal cells is higher than what it is in a rodent. So these probably are energetically a little bit expensive guys. So there must have been a pressure to somehow take down a little bit the ATP consumption. So one way to take it down is just to allow them to generate action potentials with 3 times smaller current transfer or the ion transfer, because that would actually reduce the load the cells have after the barriers to actually maintain and repair the dissipated transmembrane ion gradients. And that's where the ATP is used for. But because it comes with a high price, making the cell soma electrically slower, there has to be a secondary compensation. And we suggest that this tight association of these two features could exactly be that. Because as we saw with this pharmacological model and the model cell, that if you modulate these features, you make cell firing threshold lower, you can actually significantly shorten the input-output transformation delay. And thank you very much for your attention, and I'm happy to stop this talk to the picture of our team and our beautiful building, which was established with EU money about 5 years ago. Thank you.
Thank you. Great talk. One of the stories about why the fast-spiking interneurons can work so fast, at least in the rodent literature, is that they have calcium-permeable AMPA receptors which generate very fast EPSPs to drive them high. Do we know in the human situation that the synaptic inputs would be similarly fast?
They are not too much longer, but they are a little bit longer when we compared the decay time constant of the EPSC in connected pairs. They seem to be, mouse are tuned to be fast. And if you push them actually in all terms, they are faster in individual circuits or parameters. But humans, they have compensatory systems that makes them faster than what would they otherwise be. But I suppose they also have calcium permeable AMPA receptors in human, but we haven't. I don't think anybody has shown it yet in a functional level. No.
What you're showing now is something that I think everyone that records from human neuron noticed, that something weird is going on with the cells, and I think you solve it. But there's come a question, because when the circuit is active, majority of the input resistance is determined by the synaptic activity, not by this kind of slice type input resistance, right? Can you say something about what will be the effect, given the network activity and multiple synaptic inputs, on this kind of lowering the threshold and all that?
Obviously, I would assume this kind of Turiciano type of synaptic scaling. I mean, phenomenologically here, that the EPSCs needed to provide excitase and can be clearly shorter, smaller in amplitude, and same for iPSCs. But if I understand correctly, the point of your question is that how is the high input resistance affecting the circuitry properties in other ways, is that they can be very, very efficiently inhibited by iPSCs. So, and they, yeah, because equally, if you take an equal size iPSC and put it in these cells, it really shunts the short circuits, the soma. It's a very effective way to inhibit the